I'm Angie F. Camarista from Criminology 22. Today's tutorial video, we will tackle about the methods of the following. First is anthropometry, second is portrait parley, and the third one is forensic odontology, the fourth is forensic anthropology, the fifth is forensic serology, and the last one but not the least, DNA fingerprinting. So, let's start. So, hi guys, let's begin our discussion. Our first topic is anthropometry. What is anthropometry? Anthropometry is dealing with the measurement of the human body to determine differences in individual. Anthropometry is designed by Alphonse Bertillon. It began in 1890 and lasts approximately 20 years, being replaced by fingerprints identification in 1940. It was officially adopted by France. Anthropometry or Bertillon system have a three composition.
The first one is anthropometrical signalment, measurement of the body, head, and limbs. Limbs, it pertains to the hand, finger, and foot. And the second one is descriptive signalment, a clear and specific system of description of an individual as well as the photography of the male factors. For example, is the mug shot. And the last one is specular mark signalment by indelible mark like scare, tattooing, birthmark, mole, etc.
So guys, our second topic is portrait parley. What is portrait parley? Portrait parley can be defined as a rigorous system from verbal description of physical characteristics of the subject. It is based on the Bertillon methods of criminal identification by measurement of human body and is still practiced today to some extent. If such information given by witness, relatives, or person who is acquainted with the physical features of the person to be identified. Here is the description of the following features considered in portrait parley.
The first one is oval, oblong, round, rectangular, square, triangular, diamond, and etc. Second is skin color, very fair, white skin, light brown, moderate brown, dark brown, and etc. In hairs type, straight, wavy, curly, coils, undefined coils. And the third one is eyes, round almond, drapy, hooded, Asian, round Asian, and etc. In eye color is gray, green, brown, black, or blue. And the types of nose, ideal nose, broad tip, broad nose, serial nose. In the occupational mark, some mark develop due to the kind of occupation performed by an individual. Some example is the dishwasher, truck driver, and etc.
And our third topic is forensic odontology. What is forensic odontology? Forensic odontology is a branch of forensic science, uses the skill of the dentist in personal identification during mass calamities, sexual assault, and child abuse, to name a few. It is the proper collection, interpretation, and evaluation as well as presentation in court of dental evidence during the conduct of criminal and civil proceeding. It is the application of dentistry in legal proceeding deriving from any evidence that pertains to the teeth. And why teeth?
The first one is every human body ages in a similar manner. The teeth also follow a semi-standardized pattern. The second one is each human has an individual set of teeth, which can be traced back to established dental records to find missing individuals. And the third one is teeth is made of enamel, which is hardest tissue of the body. And the last one is teeth are source of DNA. dental pulp or a crushed tooth can provide nuclear or mitochondrial DNA that to help identify a person. The teeth base is approximately 32 teeth in adult mouth. Teeth have a different in size, shape, and root. Teeth through the years in childhood. Primary teeth sprout from milk buds and the temporary. Once they fall out, permanent teeth as the seen on the other side appear. In adulthood, permanent adult teeth came in when primary teeth fall out. They are permanent because they establish root inside the gums. The third molar come in the around the mid-teenager years. Four types of teeth. First is molar, premolar, canine, ancestors. Analyzing bite marks. First is bite marks are photographed with a scale. Second, bite marks on skin are taken over repeated intervals. Cast of impression are taken. Impression traced onto transparencies. Cause of suspect teeth are taken. Comparison between suspect cause and bite marks. The bones were interested in skull, humerus, pelvis, femur, and tibia.
So guys, let's proceed to our next topic, which is forensic anthropology. What is forensic anthropology? Forensic anthropology can be described as the analysis of human remains for the medico-legal purpose of establishing identity. Anthropologists are relatively called upon to assist in the identification of individuals who remain are severely decomposed, burned, disrupted, mutilated, or otherwise rendered difficult to recognize. And determination of sex.
Pelvis is the best bone, which is our hips, and in the first view, first is female have wider subpubic angle. Second, female have a sciatic notch, which is 90 degree, and the third one is female have a broad pelvis in back view. First is female have a wider subpubic angle. Second is female have a broad trubel like ilium. And the third is female have a flexible pubic symphysis.
Let's proceed to the next topic, which is forensic serology. First, we need to define serology. Serology is the study of bodily fluid, is called serology like blood, saliva, semen, or sweat. And forensic serology is the application of the study of blood, semen, saliva, and other body fluids to legally matter. The field generally is comprised of the detection of enzyme and antigens, as in the identification of criminal stain or blood typing and DNA typing. What is blood made of?
The solid portion of blood is made up of three principal types of cell. First is red cells, which is contain hemoglobin. The second one is white cell, which are primary cell of the immune system, and they produce antibodies. And the third one is platelets. When the start clotting process, when the solid material is removed, it leaves behind a pale yellowy, watery fluid called serum. Is visual observation followed by a positive chemical presumption and produce a visible color or light reaction. First is catalytic color test, employs the chemical oxidation of a chromogenic substance by an oxidizing agent catalyzed by the pre-series of blood. The second one is chemiluminescence and fluorescence, involve spraying a mixture on a suspect area and observe the result. And the third one is phenolphthalein, produce a bright pink color when used in the testing suspect blood. And the fourth one is the luminol. The forensic application involves spraying a mixture of luminol over an area through to contain traces of blood and 30 seconds after spraying. And the last one is fluorescein. Similar to luminol, however, it requires on alternate light sources. Would not react if the area has been previously treated for bleach. Precipitin test, the standard method uses animal serum that contains antibodies specific to human antigens react to agglutinate human blood. ABO system 1901, Karl Landsteiner recognized that all human blood was not the same. Worked out the ABO system, discovered why so many patients died after a transfusion for so apparent reason. Discovered the Rh factor in the blood on the surface of each red blood cell. Millions of characteristic chemical structure called antigens can be found.
And last but not the least, the DNA fingerprinting. What is DNA fingerprinting? DNA fingerprinting is a way to identify a certain individual rather than simply identify a species or a particular trait. A technique used by scientists to distinguish between individuals of the same species using only sample of their DNA. DNA fingerprinting was first reported by the English geneticist, which is Alec Jeffrey. DNA stands for deoxyribonucleic acid. DNA is the genetic material found within the cell nucleus of all living things. And DNA fingerprints have
Looks into DNA. DNA looks like an incredibly long twisted ladder. This shape is called a double helix. Here are the 8 steps for DNA fingerprinting. First step, isolation of DNA. DNA must be recovered from cell or tissue. Only a small amount of blood, hair, or skin needed for isolation of DNA. Step 2. Digestion of DNA. By restriction endonucleases, the DNA is cut into fragments using restriction enzyme. Each restriction enzyme cut DNA of a specific base sequence. Step 3. Electrophoretic separation of different fragments. Fragments are separated on the basis of size using a process called electrophoresis. DNA fragments are injected into wells and an electric current is applied along the gel. Step 4. Transfer DNA on nylon or nitrocellulose membrane. The DNA fragments are transferred to a nylon sheet by placing the sheet on the gel and soaking them overnight by the process shortened blot. Step 5. Probing or probe labeling. Adding radioactive or colored probes to the nylon sheet, which is complementary to target sequences. Step 6. Hybridization. Label-proof DNA should be hybridized with the complementary sequences located in nylon membranes for the detection of positive of latter. Step 7. Autoradiography. To detect the sequences in genome bound with the hybridized radioactive probe on membrane. Technique involves alignment of hybridized membrane with X-ray film. The X-ray film alter its development show multiple of bands that look like barcodes and known as DNA fingerprints. Step 8. Interpretation of band patterns. Analysis of band patterns of different individual. Comparison of position bands. Computer software are also available for the analysis of DNA fingerprints. Here are the famous cases in DNA fingerprinting. In 2020, Elizabeth Hurley used DNA fingerprinting to prove that Steve Bang was the father of her child Damian. Colin Pitchfork was the first criminal caught based on the DNA fingerprint evidence. He was arrested in 1986 for the rape and murder of two girls and was sentenced in 1988.
So guys, that's all for today. Thank you for watching and God bless you all.